This is the beginning of Unit 4, Part 2. All right, this is the continuation of Unit 11. This is Unit 11, Part 2. We're going to talk now in more detail about white blood cells. And the white blood cells, of course, are called leukocytes. There are a number of different lineages of white blood cells. There are the myeloid cells, which are made up of the granulocytes and the monocyte macrophages. And there are three different types of granulocytes, and then there is the lymphoid lineage. And you'll see more clearly in a little bit why I talk about them that way, and that's the best way to look at them, really. Okay. These white cells, they are the cells that are what are called, I guess, complete, in that they have a nucleus and they're out in the periphery, whereas the red blood cells don't have a nucleus. And they are much fewer in number than red cells. They make up about in just terms of the volume, they make up about 1% of the total blood volume. White cells can leave the bloodstream. They leave under particular situations. They can leave through the capillaries by moving between endothelial cells that line or pave the inner surface of the capillaries. And the process of moving out of the capillary into the tissue is called diapedesis. So they are quite capable of leaving the bloodstream and moving through the interstitial spaces or tissue spaces. When we want to break down the white blood cells in terms of determining what the different populations are that are present and the numbers, that's called a differential count. If the overall white cell count goes over 11,000 per cubic millimeter, then that's considered a abnormally high count and is considered to be leukocytosis, which suggests some kind of microbial infection, either bacterial or viral infection. That's a typical very early response. You'll see leukocytosis where the white blood cell count is rising. White cells in general protect you against infection. There are quite different functions between the different white blood cells, and we'll talk about their basic characteristics and functions here. The granulocytes, which are part of the myeloid lineage, the other part being monocyte macrophages. Granulocytes, there are three types: neutrophils, eosinophils, and basophils, and they can be distinguished from each other by the staining of their cytoplasmic granules. They stain differently from each other with a Wright's stain. Wright's stain is a very commonly used stain that's done on a smear, a peripheral blood smear. A smear is simply taking some blood, putting it on a glass slide and smearing it across the slide. And then you can look at it under the microscope and look at the white cells and determine the differential count. And when you do a differential count and determine the different cells, you can fairly easily distinguish between basophils, neutrophils, and eosinophils, because although they all have granules in their cytoplasm, the basophil granules are dark staining, violet stained. The eosinophils are red staining, and the neutrophils are light, very, very light, either non-staining or lilac, very light violet staining. The other thing, of course, is that you will see a multi-lobed nucleus. It is true that eosinophils often have two lobes, and that's shown in this example. But it's also true that quite often they are more than two lobes. So that is not necessarily a distinguishing characteristic. multilobed nucleus is. They are these cells, the granulocytes, are certainly larger than red cells and typically much shorter lived than red cells. As I said, they have a multilobed nucleus, and for that reason they are called polymorphonuclear cells. Poly meaning many, morpho being shape, and nuclear referring to the nucleus. So PMNs are polymorphonuclear cells. All are phagocytic, although the neutrophils are the most phagocytic, and the neutrophils are the ones that typically would arrive at a site of inflammation before any other cell, any other white cell actually. So the neutrophils are the most numerous of the white blood cells. Here are examples of typical examples of two neutrophils, seeing the multilobed nucleus. The cytoplasm is somewhat stained, but not dark violet or red. These are clearly granulocytes, neutrophils. Neutrophils are 60% plus or minus 10% of the white blood cells in the peripheral blood. The granules contain enzymes that can generate what are called a class of molecules that are called oxygen radicals. These are highly toxic and are able to kill bacteria that the cell phagocytoses. These are highly phagocytic cells, so it phagocytoses and kills the bacteria using these enzymes to generate oxygen radicals. It'll break down whatever it phagocytoses because in the granules you also have hydrolytic enzymes. Defensins are antibacterial protein that actually works quite effectively at killing off bacteria also. The eosinophils, which account for only 1 to 4% of the white blood cells, are red staining in that the granules are red staining, usually a bilobed nucleus, but it can be more lobed, more than 2 lobes. Red granules, as shown here. And the main role of this cell is to respond to parasitic worm infections. Most of the world's population, in fact, do carry parasitic worms, either flatworms or roundworm infections. And we have, in fact, evolved together with worms that parasitize us, and it's not surprising that we have a system to deal with those kind of infections more or less successfully. Most people in the world are chronically infected with parasitic worms. The eosinophils, this is a classic look, and I've shown you here an example of eosinophil that has more than 2 lobes. So don't count on that as identifying a eosinophil. A neutrophil could have 2 lobes. A basophil could have only 2 lobes in the nucleus. You should look at these. You should look for the nucleus. If it's multi-lobed, it's a granulocyte. If the granules are red, it's an eosinophil. And here are 2 good examples. Basophils they're the least common of the white blood cells, actually, all of the white blood cells. They only form about 2%, 0.5% of the peripheral blood white cells. They also have a multi-lobed, what it means here by U or S-shaped nuclei with 2 or 3 constrictions, it means basically that there's multiple lobes, and they're going to be in an S shape maybe, or a U shape. Here's multiple lobes in a U shape. Basically, you know, I actually don't like this too much. I'm going to remove that. 2 or 3 conspicuous constrictions, narrowing, so 2 or 3 fairly conspicuous lobes. All right. Now the basophils are what you have in the peripheral blood in the circulation. Once they leave the circulation and go into the tissues, they're no longer called basophils, they're called mast cells. These cells, all of them, either basophils or mast cells, contain dark purple staining, violet staining granules, and those contain a number of chemicals. The most important, I would say, is histamine. Histamine is an inflammatory compound. If the cell degranulates, releases its granules, it will trigger an inflammatory response. It can attract other white blood cells. It acts as a vasodilator, increasing the flow of blood into the area. And it will also, and it's not shown here, but it will also increase the vascular permeability. And so not only do you have more blood flow into the area, but you also have an increased permeability of the capillaries in the area, so that more fluid moves out, and therefore you can get some edema. Not easy writing with this. Increased vascular permeability. That's an important aspect of the histamine from mast cells or basophils. Okay, classic picture of a basophil. If you look carefully, you can, I would hope, clearly see that it has a multi-lobed nucleus. You can see the violet shape there, multiple violet shapes in that multi-lobed nucleus, purple staining. And they're not easy it's not easy to see the nucleus, I'll admit, but when you see it that purple, the entire cell, interior of the cell, including the cytoplasm, then and it's granular, then you really should realize that you're looking at a basophil. Okay, the other type of cells are monocytes, which are part of the myeloid lineage, the other half of the myeloid lineage, and lymphocytes, which are part of the lymphoid lineage. They are not related. Your book talks about them as non-granular white cells or agranulocytes. It's a term that's not commonly used. I wouldn't lump them together ever. I would deal with them one at a time, and that's what we're going to do. Monocytes are phagocytic cells. They are found in the peripheral blood. 5% plus or minus 2, or 4 to 8%. I guess this is 6% plus or minus 2. They are fairly larger somewhat than the other cells, but not gigantically larger. When a monocyte leaves the circulation and enters into the tissues, it's called a macrophage. It gets somewhat bigger in size. One of the characteristic features is that the nucleus is either U-shaped or kidney-shaped, and that can be very clearly, easily differentiated from other cells. These are phagocytic cells. Macrophages that are in the tissues are even more phagocytic. These monocytes are highly mobile and can leave the circulation and move towards an area of inflammation. They are attracted to the area and are very immediately upon leaving the circulation, they're called macrophages and are highly phagocytic. They help in the activation of lymphocytes. Because they are able to phagocytose foreign objects, molecules, particles, and then present them on the surface. So they'll chop it up into small bits and then present on the surface of the macrophage. They'll present that antigen to lymphocytes. And lymphocytes have receptors for antigen, for foreign molecules. and respond well to antigen that's presented by an antigen-presenting cell. So macrophages are important antigen-presenting cells. Lymphocytes is part of the lymphoid lineage of cells. They're the second most common white cell in the peripheral blood. They account for about 25% of the white cells. They have a circular, more or less, circular nucleus. They don't have a large amount of cytoplasm. There are two main types of lymphocytes. There are T lymphocytes and B lymphocytes, quite often called T cells and B cells. These lymphocytes are the cells that mediate the specific immune response, better known actually, or more correctly known as the adaptive immune response, which is highly specific. The reason it's so specific is because these cells have antigen-specific receptors on their surface. So the adaptive immune response is mediated by T cells or B cells. The T cells mediate their immune function by the cell itself, whereas the B cell is able to produce and secrete antibody molecules and will mediate their immune response through the secreted antibody. So for T cells are mediating cell-mediated immunity, and B cells are mediating antibody-mediated immunity. And together those form the basis of the adaptive immune response, which is also called the specific immune response. This is a table, this page and the next, I would guess, summarizing, more or less what I've said. There's more information. You can read it. You can look at it. Okay, we're going to talk now about production of white blood cells, or leukapheresis. We already introduced the term hematopoiesis, which is a general term for any kind of blood cell production, and erythropoiesis is specific for red cells, and leukapheresis is a term for white cell production. Just like with erythropoiesis or any of the lineages of cell production, it is all hormonally stimulated by molecules that are typically produced by other cells. Many of these cytokines, these protein hormone signals, are produced by white blood cells, but not all of them. These factors, or hematopoietic hormones, or factors, usually called factors rather than hormones, are mostly called interleukins, and the abbreviation for interleukin is IL, and there are a number of them. They're all classified by a numerical number, for example, IL-1, 2, etc., etc. It goes quite far. Another name for some of the growth factors is a better term. Hematopoietic growth factors are colony stimulating factors, or CSFs. Many of the cytokines of these growth factors are produced by macrophages and also by T cells. And some of them have been, the genes coding for them have been cloned and are produced commercially and used clinically to stimulate bone marrow production of different types of cells. This is a classic kind of chart showing overall hematopoiesis. It's leaving out, this is leukapheresis actually. It doesn't include erythropoiesis or thrombopoiesis, which is production of platelets. Here you see the hemocytoblast The stem cell is often called a pluripotential stem cell because it can lead to all of the different blood cells, including red cells and platelets. Here you see the main lineages of white blood cell production. Here's the lymphoid lineage going down. You get differentiation and proliferation, increase in number, till it produces mature lymphocytes, and that includes both T cells and B cells. And here you have the myeloid lineage, and as you know, there's two lineages. One leads to granulocytes, including the eosinophils, neutrophils, and basophils. And here you have the myeloid lineage that leads to macrophages and monocytes. So again, there's a myeloid lineage and there's a lymphoid lineage, and the myeloid lineage includes macrophage monocyte production line and granulocyte production line. And the lymphoid lineage leads to T cells and B cell production. So this is leukopoiesis from the pluripotential stem cell, the hemocytoblast. Notice I didn't go through all the names in between the most primitive cell, the hemocytoblast, and I gave you the final name of the cell. There are all kinds of names for different stages, immature stages in between, but you wouldn't have to know those. So all cells, all the cells of the blood, including the white cells, the leukocytes, originate from hemocytoblasts. These are the pluripotential stem cells. And in terms of leukopoiesis, white cell production, you have these can differentiate towards myeloid cells or lymphoid cells. And the myeloid cells can lead to development into the different granulocytes, the eosinophils, neutrophils, and basophils. Or if it follows a different line of differentiation and proliferation, depending on the signals that it's getting, cytokine signals, they'll differentiate and proliferate into monocytes. And the lymphoid lineages will produce lymphocytes. This is a lymphoid, it commits to this lymphoid direction. Okay. So you see that word committed. It's committed when it gets certain signals and it differentiates to there. It's committed to develop into these kind of cells. Once it goes in this direction, both myeloid, but different signals, then it's committed towards production of monocytes or macrophages, monocyte macrophages. And here you have commitment towards the lymphoid lineage, committed cells. What it's saying is that these cells, these intermediate types of more primitive cells, are stem cells for lymphocytes or macrophages or granulocytes. They're committed towards that lineage, and they can't go to other ones, but they're still, and they're committed because of the signals they've received in terms of soluble cytokine signals. Are there disorders? Yes. Leukemia is a tumor of white blood cell origin, and it's named according to what kind of white cell is involved. So you could have a myelocytic or lymphocytic leukemia, depending if it's myeloid in origin or lymphoid in origin. It can be acute or chronic. It could be, there are actually many different kinds. In general, I wouldn't worry about these terms. Acute leukemias typically involve more primitive, less differentiated precursor kind of cells, abnormal. You get very rapid, that's why it's called acute, proliferation of less well differentiated cells that are abnormal. They're some of the earlier lineages, and typically this is found in children. Many of these can be, they've been very successful at developing treatments for these kinds of acute leukemias, acute tumors involving white cells. The chronic ones, or chronic leukemias, are more prevalent in older people and somewhat less, slower growing and less successful in treatment, but still they do have some success. You know, this is a giant topic and I can't do it a lot of justice, but I'll give you the basics of it. With leukemia, you see abnormal, poorly differentiated white cells in the bloodstream. The bone marrow also becomes occupied by the, taken over by these abnormal tumor cells derived from different types of leukocytes. Typically, death is due to hemorrhaging because of the effect on other lineages. These people do not produce, their bone marrow is taken over by the abnormal tumor cells and they don't produce enough platelets particularly. Typically they're also more susceptible to infection because they're immunosuppressed. Treatment includes irradiation, anti-leukemic drugs, which is called by the public chemotherapy, but the public, I would say, is pretty much believes that chemotherapy refers to anti-cancer drugs, anti-neoplastic drugs, but it refers to any drug. An antibiotic is a treatment is chemotherapy. So, anti-neoplastic drug is a better agent, or drug is a better term than chemotherapy. The other type of treatment is bone marrow transplantation. This is a tumor, leukemias are tumors of the white cells, the blood, and with irradiation treatment or anti-leukemic drugs, administration of anti-leukemic drugs, quite often the rest of the bone marrow is destroyed, the stem cells are destroyed, and they need to get a bone marrow transplant from a different individual. Platelets are part of the production of platelets is called thrombopoiesis. Sometimes platelets are called, and you will see the term, sometimes they're called thrombocytes. And the production of platelets, the process of production of platelets is called thrombopoiesis. They originate also from the pluripotential stem cell, the hemocytoblasts. And there's a maturation towards a cell called megakaryocyte. Megakaryocytes are found only in the bone marrow, and the platelets bleb off. They come off of the megakaryocyte. You have an awful lot of platelets in you, but they're very, very small, so they're not that noticeable in the peripheral blood. Platelets act by forming the very first reaction, actually, to a break in a blood vessel, and they'll form a temporary platelet plug to start off the overall coagulation response to a tear in a blood vessel. Here you see the stem cell hemocytoblast. This whole process here is thrombopoiesis, and you see that there are different intermediate cells, just like with the other lineages. But the final cell that's produced, but nevertheless remains in the bone marrow, is called the megakaryocyte, and the platelets just continually bleb off of the megakaryocyte and go into the peripheral blood. During hemostasis, there are three phases that occur in rapid sequential order. The first is a vascular spasm. involving vasoconstriction, so the smooth muscle constricts around the blood vessel where the cut has occurred, and this is in response to injury. The next is platelet plug formation. The platelets will adhere to each other and form a plug. And then the third is coagulation. The clotting cascade gets initiated, the blood clots and forms a barrier against to prevent any further leakage of blood. The body looks at bleeding as an emergency and responds rapidly. So here it says platelets do not stick to each other or to the endothelial lining of blood vessels. It's true. What they adhere to, in fact, is connective tissue. You know, they wouldn't adhere to the endothelial lining of blood vessels because that's what normally lines blood vessels all over the body. That's the normal lining. It's a non-adherent kind of surface for them. They don't stick to each other. I think I said they do, but they don't. They'll stick to, essentially, they'll stick to the connective tissue that gets exposed when a blood vessel endothelial surface is damaged, because just underneath the endothelial cells is a connective tissue layer containing collagen, and platelets stick to collagen. They'll adhere to it and release their contents, which includes serotonin, thromboxane A2, and ADP. What happens then is that more platelets are attracted to the site and sticking to the collagen fibers, and a plug forms at the site and limits the loss of blood. The platelet plug formation, the continuing attraction and sticking to collagen, is limited in that there is a release of prostaglandin I2. And that is in the immediate area, and that limits the platelet plug formation to that area. You know, as the blood's flowing past, it's diluting any signals for platelets to move to that and to adhere to anything. So once you have that vascular spasm and you have the platelet plug formation, you start getting into coagulation. During coagulation, the blood plasma proteins that are clotting factors, they're called factors, but each one is a protein molecule. They are in soluble form, but they will go through an activation step and then sequentially activate the next coagulation factor in the pathway until you get to factors that become insoluble fibers. There are two pathways towards coagulation. Okay, there is the intrinsic pathway and the extrinsic pathway, and there are a number of intermediate steps to these two, and then they form a common final steps, and I'm going to go through them. I'm not going to talk about these here just yet, other than to say that on the surface of platelets that have stuck to collagen fibers have aggregated and formed a platelet plug, you do get platelet factor 3 expression. The final common steps to both the intrinsic pathway and the extrinsic pathway involve factors called prothrombin activator, prothrombin changing into thrombin, fibrinogen changing to fibrin, but we'll talk about that in a minute. This is an abbreviated, simplified diagram. It shows some of the, at least at the bottom, the common steps that are common to both the intrinsic and extrinsic pathway. Actually, the overall coagulation is more complicated than this, and there's a better diagram following this. Nevertheless, what you have when you tear a blood vessel, what you get is exposure of the collagen fibers that are just underlying the endothelial cells. There are collagen fibers in that connective tissue layer just beneath the endothelial cells, and the collagen fibers get exposed with that tear. And platelets will form a platelet plug. Platelets come in and adhere to the collagen fibers and express the platelet factor 3 on their surface. Let's move on. Okay, this is the better diagram, and although it may seem somewhat intimidating, it's not all that bad. I'll take you through it so that you can understand what it signifies. First of all, before we do that, again, there are the common endpoints of the coagulation pathway. Here is the intrinsic pathway and the extrinsic pathway on the other side. The intrinsic pathway and the extrinsic pathway are activated by different signals. The intrinsic pathway is activated by exposure of tearing of the endothelial cells that line the blood vessel that's been damaged, and exposure of the underlying connective tissue material, collagen. Platelets will adhere to that, and what happens is that there is an activation sequence of different clotting factors, all of which are proteins, and once they're activated, they have enzymatic activity. Now, when you see 12, that's referring to clotting factor 12, or coagulation factor 12. And when you see an A after the number, it means it's been activated. So here what you have is activation of factor 12, so that you get 12A, which activates 11, so that you get 11A. And then it activates factor 9, so you get 9A. And it activates factor 9 with help from 7A. Okay? And factor 9, along with platelet factor 3, will activate factor 8, and you get this activated factor 9 and factor 8 complex, and that activates 10. And so in terms of the sequence of numbers, with the intrinsic pathway activated by collagen fibers that are exposed, they're underneath the endothelial cells and they get exposed, you get platelet plug and activation of 12. So you get 12, 11, 9, 8, and 9 and 8 together will activate 10. Okay, so at this point there's the activation, there's the active factor 10. So at this point you can see now that you've hit the common point in the two activation pathways, the intrinsic and extrinsic pathway. So we have 12, 11, 9 with help from 7, and you get 8 and activated 8 and 9 here will activate factor 10. So 12, 11, 9 plus 7 activates 8, and then you get 9, 8 complex activating 10. The extrinsic pathway is activated by tissue factor. Intrinsic pathway is activated by exposure of collagen to the blood. Tissue factor is the activating signal in this case, and tissue factor comes from cells that have been injured, have been damaged, and cells are damaged when you have bleeding, right? You wouldn't have bleeding unless you had some cell injury. You wouldn't have bleeding unless you had, torn blood vessel exposing the underlying collagen, and also cells get damaged, you get exposure of tissue factor. Now, tissue factor will activate factor 7. And 7, of course, helps with activation of 9 to activate 8 here. But the complex of tissue factor and activated 7 also will activate factor 10 to form factor 10A. So in this case, we have tissue factor activating 7, and the activated 7 and tissue factor activating factor 10. So factor 10 is a common point in the activation pathway. The two pathways, intrinsic pathway and extrinsic pathway of activation and coagulation, converge on factor 10. Then you get 5 activated, and with platelet factor 3 you get prothrombin activator. Prothrombin activator activates prothrombin to form thrombin. Thrombin is active. And thrombin will activate fibrinogen. Now fibrinogen, all of these inactive molecules are soluble proteins. Fibrinogen and all the active ones that I've talked about so far are also soluble. But at this point, this is the point at which you get an insoluble product. When fibrinogen is activated, it becomes insoluble fibrin. And that also requires factor 8. So factor 8 is important in the cross-linking. Its cross-linking strengthens the fibrin strands. But prothrombin activator activates prothrombin to thrombin. Thrombin activates fibrinogen to fibrin. Fibrin is insoluble, and with factor 8 gets cross-linked and forms a really strong fibrin mesh. I realize it's not that easy or simple, but you have it. It's in front of you here. You see the 2 separate pathways. It actually makes a lot of sense how they're activated by either collagen or tissue factor or both, leading to insoluble fibrin polymer, long strands of insoluble fibrin that are cross-linked by the action of factor 8, and you get a stable blood clot. So overall, coagulation, the first phase, are the 2 activation pathways leading to prothrombin activator. All of this is initiated by tissue damaging events, a tear, cells being injured. You get a series of procoagulants that lead towards activation of factor 10. And that activated factor 10 will complex with these, platelet factor 3 and 5, to form prothrombin activator. In the next phase, prothrombin activator will activate prothrombin to form thrombin. And once you have active thrombin, and when I say active, it means it now has enzyme activity and can act on the next factor in the pathway. So phase 2 is activation of prothrombin to thrombin by the prothrombin activator. And phase 3 is thrombin acting on fibrinogen to form fibrin. These insoluble strands of fibrin form the structural basis of the clot, but and they do form a trap. It's quite good, but they are reinforced and strengthened by the action of factor 8. Okay, so fibrin in the presence of calcium will activate 8, and that helps cross-link the fibrin strands, strengthens and stabilizes the clot. Factor 8 is extremely important. Factor VIII has another name besides factor VIII. It's also called von Willebrand factor, and a deficiency of von Willebrand factor or factor VIII deficiency is a genetic type of oh dear, I've forgotten the term. Genetically based hemophilia. That's one of several different types of hemophilia that exist. Hemophilia is a genetically based disease in which the blood doesn't normally coagulate. So factor VIII deficiency is one of the more common types of hemophilia. Not that hemophilia is that common. The nice thing is that the gene coding for the protein factor VIII has been cloned, and factor VIII is available commercially, and these people can receive inactive factor VIII so that they can carry out normal blood clotting. Now, whenever you get a clot forming and the clot gets cross-linked, so you have these, you have prothrombin activator activating thrombin, activating prothrombin to thrombin, and thrombin activating fibrinogen to fibrin. You get these fibrin strands that form a long meshwork and that gets cross-linked because of the action of factor VIII. But one of the important things is that what you get after that fibrin cross-linked meshwork, it will retract. It shortens and squeezes out the remaining liquid serum from between the fibrin strands. So you get, after the clot forms, you get a process called clot formation, or clot retraction, where the strands are shortening and squeezing out the liquid serum. And that is followed by repair. Whenever you have damage, you not only want to stop the bleeding, but you also want to repair. And one of the things that platelets provide is a growth factor called platelet-derived growth factor, or PDGF, which helps stimulate the repair of the blood vessel wall. You also get some fibroblasts coming in to secrete collagen that acts as a connective tissue patch in the wall. And you also get release of another growth factor called the vascular endothelial growth factor, VEGF. These are a lot easier to remember as VEGF and PDGF. Platelet-derived growth factor, vascular endothelial growth factor. This helps endothelial cells, the cells that normally line or pave the surface of the blood vessel. It helps them to grow and multiply and restore the proper endothelial lining. Always a very important part of injury is repair, wound healing or wound repair. In this case, it's repair of the blood vessel. So you get clot retraction and repair mechanisms. The clot retraction happens quite quickly within I would say about the first hour or so after the coagulation events, but the repair takes longer. It takes several days. Now, part of that process also involves the dissolving of the clot. The clot doesn't hang around. It will get dissolved. And we're not talking scab, we're talking blood clot inside the tissues, right? We're not talking about a dried scab on the surface of your epithelium, your skin. We're talking about inside. You get a process occurring called fibrinolysis, the lysis or breaking apart of the fibrin strands. And this normally happens within about 2 days after the initial formation of those strands, and it helps to dissolve the clot. Dissolving the clot is in fact part of that whole wound repair process. Does clot retraction, or does clot formation or growth, is it limited? It has to be limited. It can't expand and fill up your whole bloodstream with clot. That would be bad. So there are mechanisms that prevent the clot from becoming too large. The first one is that the activated clotting factors, those activated proteins, get diluted in the blood flow away from the site, so they're diluted. And the other is that there are negative inhibitory feedback signals that will inhibit further activation of the clotting factors. And those negative feedback signals come from the fibrin itself and also from heparin, which is produced, it is used as a drug in the hospital, but to prevent blood clotting, but it also is produced by endothelial cells. There are a number of anticoagulation factors that can limit the extent of a blood clot. You don't want to have a clot forming and getting bigger and bigger and spreading through the bloodstream. And those negative signals include fibrin, heparin, antithrombin 3, and protein C. So normally, in a normal, uninjured blood vessel, clotting is prevented by the integrity, the complete normal paving of the endothelial cells that line the blood vessel. And those endothelial cells provide a nice smooth lining, and they prevent any platelet adhesion. They secrete heparin and prostaglandin I2, which are anticoagulant, have anticoagulant activity, and they also secrete vitamin E. So all of this is basically the surface of your blood vessels, when they're intact, is quite inhibitory towards any coagulation. Now, sometimes a clot will form in an unbroken blood vessel, and this is a problem. It will start to develop and it'll persist or stay in the unbroken blood vessel. And this kind of clot in an unbroken blood vessel is called a thrombus. And the thrombus can actually get large enough to block circulation, resulting in downstream death of tissue, because the tissue that's downstream from that blockage site can't get enough adequate circulation or perfusion, doesn't get enough oxygen, and can die. If that occurs inside a coronary artery, then you have what's called a coronary thrombosis, and the thrombus will block the blood vessel and that's providing oxygen to part of the heart muscle. Now, a really serious problem, much more serious than just thrombus formation, if you have a blood vessel, let's draw it like this. You have a blood vessel, put it in black. You have a blood vessel, and for some reason, because of some chronic disease problem, you get a thrombus forming. So you get this blood clot forming, and they always trap some red cells, so it's forming, and it can extend down, and it can be fluttering even in the stream of blood. Okay, the liquid blood is flowing past it, but sometimes the thrombus breaks off, and when it breaks off, then it is called not a thrombus but an embolus. So an embolus is a thrombus that is broken off and is free floating in the bloodstream, and it'll keep floating through the bloodstream until it gets to a blood vessel that's too narrow for it to pass. These kind of events, developing an embolus, can be extremely serious, have extremely serious consequences, because wherever it ends up sticking and blocking a blood vessel, it will cause downstream tissue injury and death, because downstream of the blockage site you will have hypoxia of the tissue, and you can get death of the tissue downstream. If it blocks in the lungs, if it travels in a vein back to the heart and it gets pumped up to the lungs, and that's the first capillary bed in the pulmonary circulation, so it could block a blood vessel in the lungs. It's called then a pulmonary embolus. That's serious. Much worse is if the embolus flows into the brain and blocks a blood vessel in the brain. That's a cerebral embolus, but that is also the basis of a stroke. A stroke essentially is a lack of circulation, hypoxia to an area in the brain downstream of a blockage or bleeding, and loss of that tissue. The tissue dies. Can undesirable clots like thrombi, emboli be prevented? Yes, there are a number of drugs that prevent it. Aspirin, which has inhibitory effects on thromboxane A2, so it can prevent undesirable clotting. Heparin, which, yes, is produced normally by the endothelial cells lining the bloodstream, but is also available as a drug, a pharmaceutical agent. It can be used as an anticoagulant. And warfarin. Warfarin is the generic name for a drug which is called, when it's used on humans, it is called Coumadin. And many people are on Coumadin therapy to prevent unwanted or inappropriate formation of blood clots and coagulation in their bloodstream. Coumadin. Coumadin is generically known as warfarin, and I can tell you that warfarin is what you have in rat poison because they mix the rat poison in with some food for the rats to eat, and then they become anticoagulated and they'll bleed to death. You are constantly getting tiny little bleeds. You or rats, it doesn't matter. You always have tiny little bleeds occurring just from day-to-day motion and action, and they're prevented and stopped immediately by vascular spasm and platelet plug formation and small little clots sometimes. But without that, if you take strong anticoagulants, it is possible to get bleeding if you take an overdose of it as a toxin, a poison. That's what they use it as a rat poison. But it is used as a drug agent very carefully, this or heparin, in very carefully dosed amounts to make sure that the clotting time, the coagulability, let's say the coagulation potential of your plasma is not too high. And that's the end of Unit 4, Part 2.